firstly, I can welcome you to this interview, and please tell us a little bit about your laboratory and the scope of your research.
Thank you for inviting me. My name is Deb Ferrington. I'm located at the University of Minnesota in the Department of Ophthalmology and Visual Neurosciences. And at Minnesota, I have been working with the Minnesota Lines Eye Bank for about 17 years now. And that tissue that we get from the eye bank is how we are investigating age-related macular degeneration. What we have found, probably our most important discovery, is that the mitochondria in the retinal pigment epithelial cells is losing function very early in the disease. In fact, a long time before vision loss occurs. And so we've really been trying to understand that process at the molecular level to mostly provide some information to pharmaceutical companies to go after drug targets.
Tell us about that process that you've been undergoing.
So we started off doing proteomics. And we have a special way to evaluate the stage of the disease for the donors. It's a special system that was developed from the ARADS classification system. So we can tell when a donor has no disease, very early disease, intermediate disease, and late disease. So that's been a huge advantage for our particular investigative approach. And from that process, we were able to look at the proteins and how they change with the disease. And that's how we discovered that the mitochondria was affected early in the disease. And then we also use other biological or biochemical techniques to look at mitochondrial DNA damage. And again, that confirmed that the mitochondria is damaged early in that process. So those are the big discoveries that we've made. And more recently, we've started to culture retinal pigment epithelial cells from our donor tissue. And that's been very exciting because what we're seeing in cultures would be what we would predict based on our previous work in the tissue. And that is mitochondrial function is lower, depressed in donors that had AMD. So we have a really nice system now to go in and really investigate macular degeneration at the cellular level.
What measures of mitochondrial function are you documenting?
Okay, so we have mostly been using an equipment called Seahorse, which allows us to actually grow the cells in a dish and then test the function while the cells are still, the mitochondrial function while the cells are still alive and in place. There's lots of different ways to do it, but this is one way that's been very effective for us. And we can measure things like how much ATP the cell is making, what the maximal respiration of the cell is, what's the basal respiration. And what we've done is actually go in and apply different types of drugs to see if we could improve mitochondrial function. And that was part of the research that I presented at ARVO, is looking at how do we improve mitochondrial function that, again, might lead to some therapeutic interventions.
So switching to presentations here at ARVO 2017, tell us a little bit about what you presented.
Okay. Well, one of the presentations was about just exactly what I was saying. We're using the culture system now as a way to test for response to drugs, and that's been very successful. There's a few FDA-approved drugs that we repurposed and looked at the way that it affected the mitochondria. So we've used drugs that increased biogenesis of the mitochondria, increased autophagy, which would get rid of damaged mitochondria, and then also a drug that would protect the mitochondria. In all instances, they did improve mitochondrial function. But the most interesting thing is that the cells from different donors responded differently. So some drugs were good in one donor and less good in another, and so forth. So that gets to another thing that I think is important. I think we need to move in the direction of personalized medicine. I think the genetic background of the individual is going to determine their response to drugs. So one drug is not going to fit everybody.
Which agents did you test?
So we tested rapamycin. We tested a compound called pyrrolquinones, quinolone, quinon, PQQ. You can get it on the internet, and I think it's advertised as protects and energizes cells. And then the other drug that we tested was N-acetylcysteine, which has been around for a long, long time. That also improved mitochondrial function. And usually it took 24 to 48 hours for these drugs to actually have an impact on mitochondrial function. But again, it depended on which cells, what donor was being tested.
But you mentioned there was a difference in the therapeutic efficacy, or the impact rather, of these medications. Are you going further and looking at the genomics of those individual samples now?
So I have partnered with Anand Swarup, who is at the National Eye Institute, and he has about 500 samples from me. So this has been accumulating over 17 years. And he is doing RNA-seq on all of these donors that have been graded for macular degeneration. And in the end, he's going to have all of this information online for everyone to access. And I'm not sure exactly when that's going to happen, but that is the plan in place, so that everybody will be able to dive into that huge amount of data he's generating and see what they can come up with.
Sounds like it's going to be a wonderful resource for researchers around the world.
That's what the plan is. That's why I partnered with Anand, because he has the resources to be able to really do this right, and I'm really excited about it.
Where do you see this moving 1 year and 5 years from now?
Boy, that's a good question. I think we have to continue to develop models for testing different hypotheses about macular degeneration, and whether it's from donor tissue like I'm doing, or other models that are available. There are some animal models that will test specific ideas. But I think also ultimately we have to move towards personalized medicine. And another thing that's happening in my lab that I didn't present here at the meeting, we've developed iPS cells, and we've made them RPE, with the idea of, again, starting in the clinic, taking, actually we've taken used conjunctival biopsies, make them iPS cells, RPE, and so from a patient, then we could test, say, 10 different compounds that might improve function, and then select the best compounds for that particular patient. So that's the direction my laboratory is moving, and I think there's a lot of people moving in that direction also. The amount of variability in response to drugs, I think, is well known, and I think we have to address that.
So you see this in the clinical setting being a required testing process before personalizing therapy?
I think so. I think that's the direction things are going. So in the next, I would say in the next 5 years, that may be standard protocol in a lot of different institutes.
So tell us about some of the other areas that you're working on in your laboratory and collaborating on with colleagues.
So the other area that my lab is focused on is looking at protein degradation. And that actually is another component, I think, that's important for macular degeneration. We've been working on the proteasome for a large number of years now. And specifically, we're looking at a specific subtype of the proteasome called the immunoproteasome. Normally, it's present in high abundance in immune cells, and it's part of immune surveillance, but it's present in the retina, it's present in the retinal pigment epithelial cells. It's upregulated with cell stress, it's upregulated with macular degeneration. I think that particular proteasome subtype is part of the stress response. And so that's what we've been investigating. We have animal models that we've been using to investigate that particular idea. And so that might be another target for drug therapy of some sort.
Are there any other areas that you'd like to spotlight?
I think the important thing that you get from ARVO and meetings like ARVO is the coming together of scientists from many different disciplines who start to talk and interact, listen to each other's presentations. That's really valuable, but it's also the clinical component that's brought in too. And I think it's the marriage between the different fields and between the clinicians and basic scientists that's going to be really valuable in understanding this disease and moving forward with a treatment, finding a treatment. And the other thing that's important at ARVO is there's a lot of pharmaceutical companies that come here, again, looking for ideas, new ideas, seeing who to partner with as far as people from academia or clinical people. And I think that is the value in ARVO, because we're all under one roof for a whole week, and we get to see each other and talk. And I think that's the way we have to go. It's a multidisciplinary approach that it's going to take. A single lab is not going to be able to solve this problem. But if we all work together, I think that's going to be very valuable.
Finally, looking at what are the implications for patients in terms of translational medicine. From your particular line of research with regard to personalized medicine in dealing with mitochondrial dysfunction, what can you tell patients might be down the road for them in terms of therapies?
Well, that's a little beyond some of my expertise, but what I could predict is we will go in the direction of personalized medicine. I think it's not just for ophthalmology, but for a lot of different diseases. Already cancer has gone that way, very much so. I think there's a lot of people working on this disease, and there's many different aspects of the disease that are being targeted right now, and I think that's the right way to go. We should not put all of our eggs in one basket. Look at every option, because again, I think the genetics has shown that this is not a single pathway that leads to the clinical phenotype. It's multiple ways that an individual, something goes wrong, and they end up with macular degeneration. So that tells me there's going to have to be multiple treatments, and somehow we've got to figure out in the patient what's going to be the best treatment. But as long as we have multiple options, then I think therapy is going to be possible. And I think there's a lot of hope. So I'm excited about it. I think we've made huge progress in the last 5 years, and I think we're going to continue to accelerate in that progress.
Fantastic. Professor, thank you so much for this great insights, and we look forward to watching, listening, and learning as your research proceeds.
Thank you very much.